Okay. Hi, everybody. It's really great to see so many faces here. So I think most people know me. I am Gabi Conacher, and I am mom to Elliot, who's 11, who has an R850Q mutation, kind of one of these really complex mutations that's pretty profound. And I'm one of the co-founders of the International SCN8A Alliance with Michael Hammer, who I think everybody probably knows, and Jayetta Hecker, who's my mom, who's on here somewhere. And we started the International SCN8A Alliance. Well, officially it was called the Alliance in 2019, I believe, but we've been working for over 11 years now, as a team. When my son got his diagnosis in 2014, or yeah, 2013, 14, I can't remember. The years are all blurred together. Michael was one of the first people I found because his lab is the one that discovered that SCN8A can cause epilepsy in children. Discovered that in his daughter. So, he was one of the first people I contacted, and we've been working together ever since. The three of us as a team, and our work has been expanding over the last 4 years or so, really. And I think there's really an exciting trajectory right now for SCN8A. I'll just give a quick overview, and then I know Michael's going to talk more substantively about science and where we are with SCN8A. But I will tell you honestly that when we got Elliot's diagnosis, I think there were about 8 published cases in the world, and they didn't know anything about SCN8A, really, what it was really going to look like for kids with SCN8A. And we've learned that it's much more complex than anybody ever would have imagined. There's kids who are running and talking and going to school at grade level, and there's kids who have really, really complex challenges with almost every aspect of their lives. So we know that there's this spectrum, but we are all SCN8A, right? We all have challenges because of SCN8A. And our mission has always been to advance the science of SCN8A, to learn as much as we can about the disorder, the complexities of it, all of SCN8A, no matter how it looks, and really start to get us closer to better treatments. And ultimately, we are working towards a cure. And, we've had incredible progress. The number of publications that are coming out with SCN8A are, really growing over the years. We have a lot of partnerships that we are managing with a bunch of researchers and clinicians and families, family organizations around the world. And, I can continue to dig into more of that, and we can all do that in conversation. So I think maybe what we'll do is we'll start kind of with an update on the science and the research. And then we definitely want to hear from all of you and connect and chat. So I think we'll start there. Michael, are you good to jump in? Or I know we can have a conversation too about where we're headed, but
It's up to you guys. I think we should do a check-in at some point, but I've got about 10 or 12 slides that won't take that long to go through. Because my research up to today is entirely about the roadmap.
right. And are you want to give the background on that and where we're headed, or do you want me to talk for a minute about that?
Yeah, I'm going to talk a little bit about that, and you can chime in. And I'm making my coffee still because it's just turning to noon here. Yeah, so let me share my slides, and we can start from there. So today I'm going to really limit the discussion, my slides, to this SCN8A Global Research Roadmap. There are lots of exciting things to report in the world of SCN8A research that I'm not going to cover here. We often have updates on those, whether it's research on mouse models or on different advances in improving therapies or understanding different mechanisms of SCN8A disease. And those are all very exciting and important. And we will continue to report on that as we go forward. But in 1 month, we're going to have this meeting in Boston, Massachusetts. It's going to be a very jam-packed 4-day weekend or 3-day weekend with at least 40 participants in person who will be representing families, clinicians, and researchers and industry. And we, as you all know, we've put out a request for you guys to fill out our caregiver survey. We want to know what your priorities are for research. And we're going to compare your priorities for research and what the biggest problems are we need to tackle to make the most progress in finding a cure, finding treatments that will improve the quality of life for our loved ones. But we want to know what the clinicians think, what the researchers think, and where industry is at. So we will have a presentation on the results of the surveys for each of those stakeholders. There's 4 stakeholders. I will cover the caregiver research survey, and others that we've asked to step up will cover the research update, the clinical update, and the industry. And then once we've presented those different points of view where we've asked people to identify gaps in their knowledge in each of these 4 areas, then we're going to, my job then will be to try and integrate across the different stakeholders' answers in the survey. And it's very interesting already. I can see very interesting parallels and also interesting differences. So we as caregivers have our priorities, and I think our priorities should be first. But we are limited by what the clinicians can do and what the researchers can do. So it's an interactive, iterative process, and that's what the meeting in Boston will be about. At the end of the meeting, we hope to boil down all of your priorities into a manageable number of projects that we want to support going forward. So it's either going to be a huge grand challenge, or it's going to be lots of little projects that maybe each of the different alliance global groups will support individually. I'd like to see something that's synergistic so that we actually encourage researchers and clinicians and industry to work together to solve the biggest problems in front of us. What are the biggest problems that we need to solve? One, for example, would be if you have a new magic molecule or a gene therapy that actually fixes the gene, did you cure the disease? That's like the elephant in the room. So all these stakeholders are working on, whether they're researchers, industry, or some clinicians working on better treatments. If we find the magic treatment, it's going to take some time, obviously. So we've asked people to invest, whether long-term or short-term. That's one of the critical issues. If we can fix the disease and your loved one has had seizures for 6 months or 10 years, can we actually see that treatment is going to reverse the disease, bring it back more to neurotypical? And nobody knows the answer to that. So I like to think of it as we've got to hedge our bets. We've got to think about what if we don't find the magic cure? What if the new treatments don't fully cure the disease? What are we going to do in that case? And I think we need to look at other angles, not just molecular and gene therapies, but there's neuromodulation and neurostimulation, and there's all these different fields that we're not that aware of in SCN8A. And so another point of the meeting is to find these gaps. What can we do given our community of expertise, given our likely ability to fund these efforts, and say, what other resources do we need? What other stakeholders or expertise do we need to bring into the field of SCN8A to tackle the largest problems that we face? So today I want to talk about the largest problems that, if solved, will leapfrog us ahead, not incrementally move us forward in small steps as we are already doing, but to leapfrog us to the future of, having better treatments, of having better quality of life. So that's what I want to cover today. And it's a brief survey that most of you have filled out, and I think we've gotten over almost 150 responses. So we're very pleased, and I think we have a good pulse of what you guys all believe are the priorities. So we're going to talk about that, and then we can discuss them a little more in depth here as well, and find out your personal opinions and your advocacy for various things. And of course, please ask any questions about what this all means at any point of the way. So the other job of us in the meeting in Boston, we will have many families with us. And our job as researchers and clinicians and industry folks is to explain the science, the more technological stuff to families to make sure everyone understands, everyone's on the same page. So we want to do that here as well. So as you know, we have begun this international SCN8A Alliance, which has really stimulated a huge amount of progress. We're very proud of the work that we've been able to do over the last several years by shaking hands between Wishes for Elliott, the Shea Hammer Research Foundation, and then, really looking at all the stakeholders, holding families in the center of everything we do. And then moving on to our Global Leaders Alliance, and you can see here, this isn't very large, but we have groups in Italy, Spain, Portugal, all over the world that have formed and have their own localized efforts. So we want to make sure we are the umbrella over all those local efforts to make sure we're working together in concert, not stepping on each other's toes, not duplicating effort. And we've had great buy-in and great progress as a result. This on the left just shows you some of the science and some of the great achievements that we have achieved as a group of partners across the world. And Gabi, maybe you can put the link to, I don't know if that paper is actually available, but we wrote a paper from a family's point of view about the history of SCN8A and how we got to where we are. The other major achievement in the last 3 years has been the production of a consensus, a guideline for clinicians to know this disease and how to treat it, how to diagnose, how to treat it, what the prognoses are, what are the different phenotypic subgroups, and what are the different comorbidities. And I'm not going to discuss that today, but you can go to the SCN8A Alliance. It's chock full of information, almost overwhelmingly so. So I know that Gabi and on the team on the website are working to simplify that so that families can skip through and just see the highlights and the clinicians can dig into more of the details. So let's turn now to the survey itself, okay? This is going to show you the geographic origins of the people who participated in this particular survey. And I'll say that it was fun watching these numbers come up over the last few years. We always have a USA bias. Maybe that's because we started in the USA and maybe because it's an English-based thing, but we've really made a lot of efforts to make it international and many languages so that language is not a barrier to any of our progress. And at first, the Spanish SCN8A group España stepped up and they got their local families to fill out the survey. And so they were number 2. And then within the last week, Brazil just overtook Spain because the Brazilian family groups must have done some big advocacy. And suddenly a bunch of caregiver surveys came in Portuguese from Brazil. And then we have Italian, Colombia in South America, a surprise. Lots of people in Colombia who are participating and are stoked to help us out here. The UK, Canada, Poland, Australia, Netherlands, and on and on. You can see there are multiple families in each of these regions. And then the singletons out to the right. And we know there's more families out there, but I think that like I said, I've kind of watched the survey and what the results have been, and they've shifted a little bit, but they're starting to really stabilize. So any new families that come, we want to hear your voice. But I think the results I present today will be relatively representative of where you all are at. And I really want to have some discussion around it. So the first thing we asked was, what are your top 5 challenges that are most important to improve your child's quality of life? So what challenge do you face now? And where this gives us a hint of where you would like to see progress in the future. And there's so many different challenges. And I'm going to quickly just kind of go through the most common ones, because we need to communicate this to the researchers and the clinicians so they know, well, this is what we suffer from, and we want to find improvements in our quality of life with regard to these challenges. So over here you have speech, and both receptive and expressive speech are a common problem. Now, I will note that some of us today on the call have variants that produce more of a gain of function effect, more on the seizure end, more on the and some of you have loss of function variants that more on the behavioral end and developmental delays. Everyone suffers from developmental delays. It's a disease that can be characterized as a developmental encephalopathy at base, and that all these other things get added on, including seizures and an epileptic encephalopathy. So that's the complexity of the disease that we're learning about mechanistically. And our goal is to try and get these folks to think about, do we treat these things the same way, differently? Do we have different treatments for loss of function and gain of function? We don't know the answers, but we think those are important questions. So some of the things that you'll see here are more representative of the gain or the loss. And I think speech is common to both. Then we have things like seizures, which really is the most common challenge. Cognition and hypotonia, as well as motor function. So gross motor is more commonly a problem than fine motor, although I think, it depends how you ask the question. We asked it specifically, challenges with arm and legs and trunk versus challenges with fingers and wrists. So depending how you answer the question, you don't know exactly how you're going to get the answer back, but this is the transparency. I want to make sure we understand the way the question was asked can influence the way it was answered. Then if you kind of look over here, GI issues, eating safely and feeding independently, and GI dysfunction are here. I think these are more a problem with the more severe end of the spectrum. So you're getting a signal coming from that end of the spectrum there. And then sleep is a big issue for everyone. I was curious to see that falling or staying asleep was a bigger issue than sleepiness during the day. You might think that not sleeping well might make you sleepy in the day, but apparently not so much. So sleep at night is the problem, not too much sleep in the day. And then here we have the signal coming through from more on the loss of function and the behavioral self-regulation, the autism spectrum and features of autism here, which are common to both loss and gain, but much more so on the loss side. So I think we're getting kind of a picture of all the challenges from all the people with different manifestations of the disease. Any questions there? Anything that's surprising?
Where does cortical dysplasia fit in this?
We have cortical vision impairment. Is that what you, oh no, you mean like the cortical dysplasia like you would see with areas of the brain that have images on the MRI? Is that what you mean?
Yes.
That's, we didn't ask about that, Jennifer, but we do have some families that have reported that. One family had another variant at DEPDC, which is known to cause cortical dysplasia. And I always was wondering if that was the factor. Do you have evidence that in your child that's an issue?
Yes, it is diagnosed.
Okay, so you can see something on the MRI.
It's an MRI, yes.
Yeah,
Okay.
I think we're getting, I've heard a few families mention that so far, and that's something we could start looking into more. As long as if we have genetic reports from people, like Michael mentioned, there's this DEPCD that is associated with cortical dysplasia. And so we can start to tease apart whether it's related to SCN8A or some other genetic mutations or, anomalies that might account for that.
Definitely. I'd like to know more, Jennifer. So we'll be in touch about that.
So maybe, if people can, if you want to share any information about MRIs or, reports or genetic reports, I'll put an email address in the chat where you can send those to Michael so he can add them to our database and we can try to start learning more about that.
Absolutely. All right. This is kind of a basic question. Which is a bigger research priority, improving seizure control or improving non-seizure outcomes? As I just explained, there are many non-seizure outcomes. Most of the things shown on the previous slide were non-seizure outcomes. And obviously we want those things to improve. So, I mean, it's 60-30, 60-40 right now, improving seizure outcome versus improving non-seizure outcomes. I think that's fair. And I think when we talk to the laboratory researchers and the clinicians, Their challenges are going to be on those non-seizure outcomes. What do we do to improve them? How do we improve hypotonia? How do we improve some of these behavioral issues? We don't really know. We're hoping that controlling seizures will help control some of these things, but some people don't even have seizures. So this is an interesting snapshot of what the community thinks in general. Okay, then we asked, in terms of controlling seizures, now we're just going to focus on the seizure end of the spectrum. Please rank the following areas of research. What do you think is the most important research? And the first winner is new, more effective anti-seizure medications with fewer side effects. That's not a big surprise. But following that would be better understanding of the efficacy of combinations of anti-seizure medications. So most of us are not on a single medication. Most of us are on a combination of medications. And do clinicians have any sense of which combinations, much less which best single medication they can prescribe? Do they have any information that would help them understand, let's say, adding different medications to synergize the one they're giving you? Which one would they take off? Which one would they wean? Stuff like that. So we have another question about polypharmacy later, but you'll see. Right now, the third one, which third tied with drug repurposing and off-label drugs. So could we get better control with off-label anti-seizure drugs that are working for other epilepsies like fenfluramine and cenobamate? I think there's a lot of promise in that. And I think the answers reflect that the families are aware that there could be other drugs that can come into our repertoire and really make a difference. And drug repurposing is a way of taking drugs that are already FDA approved for another indication. For example, hypertension. Are there drugs that treat hypertension that will also treat epilepsy? That's a passion of my research. And we have an answer to that in our mouse model, that we have successfully repurposed a hypertension drug in our mouse model of SCN8A. And it's really shown some great improvements. We've got a publication on the way. And once that paper is published, I am going to work to try and get a small clinical trial on this repurposed drug. I think there's a lot of opportunity in that area, and people just don't know about it. So it's important to bring it up. And then, 7% say this is not applicable. So again, these are probably families that said, seizures aren't a real issue for me. I think it's probably a bigger number than 7%, but that's where the not applicable came in. Any surprises here? Any suggestions? If you didn't participate, would you agree or disagree with the way these things are ranked? Open it up to any feedback. Okay. Maybe you all agree with this ranking.
I have a comment.
Yeah. Oh, please.
And I guess it's maybe one of the talking about off-label. Like I guess sometimes it's difficult to tell whether or not a symptom is like a manifestation of SCN8A, whether it is a side effect of a medication, or whether it's another comorbidity in and of itself. And so, I think any medications that we could look into that could kind of address the fact that sometimes those things are so intermingled that it may not even matter, if we can alleviate the symptoms. So, I guess maybe that falls into the drug repurposing category, but I almost feel like it's trying to figure out off-label uses that may be more effective for multiple symptoms or comorbidities.
Yeah, that's a great question. I don't think we know. Again, like, what's the relationship between seizures and these other symptoms? Do these other symptoms have their own independent mechanism that is unrelated to seizures? This is the mystery of SCN8A. It's such an important gene, not only in the brain, but in the peripheral nervous system, that we need the researchers to step it up and tell us if SCN8A is affecting other organs than the brain. And that's really going to be one of the highlighted areas, I think, in the meeting, is to push that idea. So it's an area where we know little, and it's a huge gap. I totally agree with you, Suzanne. So, but when we speak with each other in these meetings, I always love to hear what these symptoms are that you're seeing and when they arose. Was it in response to adding a new medication? Was it in response to something to do with seizures or not? And then we just make note of it, and then we try and communicate that to the other stakeholders. So it's a really important area to discuss, given how many of those challenges that we are suffering from. Here's seizures. Everything else is a non-seizure outcome, although we don't know what the relationship is to seizures and how SCN8A is impacting these things. So I think that's one of the largest gaps and one of the biggest problems we need to solve. I think, Jennifer, did you have your hand up? Or did we cover it already? Okay, we'll go to the next one. Then we asked, are there any potential interventions or treatments that you believe warrant further investigation? This is kind of like going a little bit deeper and further, like what are the alternative treatments? And surprisingly, when we suggested that more investigation should be for how alternative therapies might help, like CBD, a lot of you are interested in that. And a lot of you are interested in the combinations of existing anti-seizure medications. Both make good sense. I was surprised that the alternative therapies was more a priority for you all than the existing medication combination. So that tells you, in my opinion, it tells me that people are kind of desperate, knowing that the existing medications, no matter what combinations they're in, are just not doing it, are not enough. And so we want to know more about other things that we can do that won't have serious side effects. Then the third and fourth are off-label meds, like we just discussed, and medications specifically for disruptive behavior. Because so many folks suffer from the loss of function side. That's our biggest sort of growing pool in our population, is more and more people are finding out they have loss of function. It seems to me that their needs are not being met at all. And so many of those families probably voted for disruptive behavior meds because nothing's really working there either. So, and then maybe surprisingly, but VNS and keto were not the things you guys were pushing for. I don't know how many people have tried vagus nerve stimulation. I'd say a larger number have tried the keto diet. And in my impression is it tends to work okay for a while, but it's hard to manage and hard to continue with. Sometimes it stops working. And we don't know if that's because, we've lost the ketosis because sugars were eaten, or, just managing that whole thing is difficult. So I think there's more interest and hope in alternative therapies other than VNS and keto, is what you guys are all saying.
We are being spoken to about being referred for VNS implant. I feel as though there's not that much known about it for how successful it is with SCN8A. So if there is anything that you guys know, or we could discuss a bit more in detail for us then talking with our neurology team, that would be really helpful.
Yeah. We could do a deeper dive in the registry data. When we did look last, we didn't see, it was like a dozen that have tried it. It was not a large number. And it seemed like in some cases it was game changing. It was wonderful. And in other cases it wasn't. So
okay.
I think we'd have to look into more deeply. A, we need more data, and B, like why did some people do well and others didn't?
Yeah.
The vagus nerve is a large nerve that connects your gut to your brain, and they put the stimulator somewhere in your upper chest or neck area, and it's supposed to be able to stimulate the vagus nerve during a period of excitability, or allow you to use a magnet or something to turn it on or off. I'm not familiar with the latest technology, but it's supposed to interrupt the onset of the seizure. But it may have other effects that are beneficial, given that the gut is so involved with SCN8A. Perhaps VNS has some effect there as well.
Okay.
But yeah, we should look into that more, Harriet, and see if we can dig out some more information so that you can go to your clinicians and have a little more information. They
Yeah.
they probably know more than we do, but again, with respect to SCN8A, I'm not sure. You can ask if they've ever seen any benefits in patients with SCN8A.
Yeah, lovely. Thank you.
Michael,
Yeah.
you also had a question in the chat about what you mean about that SCN8A is expressed in the peripheral nervous system. And what's that? What's the difference between that and the central nervous system?
Yeah, so the nervous system is roughly split up into two major sections. The central nervous system is the brain and the spinal cord, and the nerves that come out of this down the spine into the various parts of your body and organs, that's the peripheral nervous system. So brain nerves that innervate within the brain or in the spine, and then nerves that come off the spine into organs, and they even become sensory neurons in your fingers, your toes, your legs, and then your heart and your organs. So the question, we know SCN8A is expressed in those peripheral neurons, peripheral with respect to the central, and we don't know to what extent a mutation in SCN8A affects the peripheral. We know, here's some things we can guess though. We have a high issue with pain tolerance. It's actually more tolerant than less tolerant. And we have issues with autonomic issues with temperature regulation and things like that. Even in the GI, I was saying, the vagus nerve comes out and innervates the gut. Sort of like an aberrant expression of SCN8A in those places of the body surely have something to do with these comorbidities, but we don't know to what extent. The research is lacking on some of that. There are other ion channels like SCN9A that directly are involved in the peripheral nervous system and pain sensation. And there are individuals with mutations in that gene where they don't feel pain or they feel pain too much. And so SCN8A crosses over a little bit into that category slightly. And a lot of these other ion channels do interact. So that's another complex question for researchers, is how do these different channels interact with each other? And so when one's malfunctioning, does another one step up? How do they cross over and all that? But that gets into some fairly technical research stuff. Anything else? We can go on to the next one then. So then we wanted to know, assume you're in charge of distributing limited research funding. Which areas of research is your top, which area of research is your top priority? New anti-seizure medications, which we feel can come on board soon, relatively soon, whether it's an off-label drug or a purpose drug, or let's say Neurocrine Praxis's drug. Still, I mean, we're looking at some time out, but we still consider that short term, or gene therapies. And 3/4 of you say gene therapies are worth, I would give my money to gene therapies, realizing that it's longer term. We're talking years out. So it's a trade-off. It's like, ideally you want a portfolio where you have both short-term and long-term investments. But if you're really pushed hard, 3/4 of you would say, oh, I'm going to give my money to the gene therapies. And 1% said other. So how does that sit with everyone? Does that surprise you, or is there any questioning around that particular finding? Does it seem reasonable? All right. It's okay if you don't have any response to, or, any discussion around this. I frankly, I was a little bit surprised. I thought people would be more impatient. But I think we have to temper our expectations. That's the other thing we should be aware of, that gene therapies sound great, and it's the big hubbub in all the research quarters, and everyone at the NIH and everywhere else is, they want to jump on something that's going to be a technological tour de force, if you will. But we have to realize that it's longer term, and we don't know. The basic big question is, if we successfully treat the gene, do we treat the disease? So I like to look at the big picture and see what the risks and benefits of going forward in any of these directions might be. Then we asked, rank the importance of the following research to understand the mechanism of the disease. Now we're just talking about the mechanism. So a lot of the laboratory researchers are working on disease mechanism. We know that we need to understand how the disease works at the cellular and molecular level in order to come up with new therapies and new therapeutic targets. So these were all things that affect mechanism. And the winner, but not by much, it just nosed out the others, is how your genetic variant specifically affects the phenotype or your disease severity. And I think that's an important question myself. And we already know that genetic variants, particular variants, do show certain patterns in terms of how they express, whether it's more severe or less severe, or certain effects of the variant. Like there's certain gene variants that are associated with infantile spasms more than others. There are certain gene variants that are associated with more severe outcomes and less severe outcomes. And so I think that's an important question, but you notice that it's not hugely much more important than understanding just the basic mechanism of how seizures start and continue. So why do seizures come and go? Why do they continue? And we don't know the mechanism of that. That's something my lab is interested in. We think it has something to do with the blood-brain barrier and the seizure threshold. Everyone, even neurotypical people, have a seizure threshold. Any one of us could have a seizure at any time. It's just that most of us, our seizure threshold is high. It's hard to achieve that boundary, so we don't have seizures. But in our case, obviously, for SCN8A, even loss of function, many have a seizure threshold that's lowered, and we don't know why that is. So that's an overall sort of large-scale effect, seizure threshold. We don't know if it has, what does SCN8A have to do with that? We know it encourages neurons to fire. And so mutations in SCN8A lead to more firing on excitatory neurons, or loss of function, less firing. But what does that have to do with the neighboring neurons, the circuitry of the brain regions that are affected, the whole brain? I would argue from just basic principles that it's a complex matter. It's not just related to SCN8A, but it's related to a lot of other cellular and molecular features of the brain that may differ among different individuals. And one is the blood-brain barrier, which the disruption of the blood-brain barrier, which is the part of your blood system that protects your brain from getting toxic material in your peripheral circulatory system versus your central circulatory system, if you will. There's a barrier there. There's a boundary. So if you go out and do something with chemicals or food or smoke or whatever you do, the environment can affect the things that are floating around in your bloodstream, and we want to protect your brain from some of those. We want to feed your brain food, but we don't want to give your brain toxins. So we think that, and we've shown in our mouse model, that the blood-brain barrier is disrupted even before seizures start, which means that SCN8A is affecting what's getting into your brain. And if certain kinds of molecules that are normally in your bloodstream, like albumin or certain blood cells, get into your brain, that can cause a seizure right off the bat. So we know that's involved, but we don't have any good treatments yet. Although the drug that I talked about, the repurposed drug that I'm testing, does protect the blood-brain barrier to some degree. So are there medications that can protect your blood-brain barrier that will help increase your seizure threshold? And I want to know if we have the expertise in our research community and our clinical community to be able to address things beyond the ion channel. The ion channel is important. Most of the research is directed at SCN8A and NaV1.6 channel, and trying to correct it. But I'm hedging my bets and saying there are other factors we need to think about in case fixing the channel doesn't fix the disease. Another issue that is more global and has to do with seizure mechanisms is inflammation. We know that when our children have disease infections, or when they're teething or other inflammatory processes are occurring, that can reduce your seizure threshold. So why? Why is that? And are there medications or treatments or strategies we can use to decrease the effect of inflammation, or at least know when it's going to be a problem? And then really right in there was non-seizure outcomes. This is obviously super important. The mechanism, how drugs, so now specifically looking at drugs, why they work, why they fail. Can we predict when someone's going to be resistant to a certain drug? There's a mechanism lying in there somewhere. And then SUDEP was tied for understanding the mechanisms of SUDEP. So I would say sleep difficulties, all of these were very important. And it was difficult for you guys to separate them out into a winner and loser situation. But I think the bottom line here is they're all important, and maybe some of them are slightly more a priority than others. Any questions or
I have
comments?
a question. Has there been any research done on like the relationship between diet, not keto diet, but like anti-inflammatory diets? I mean, I'm thinking about like multiple, like MS, where you have like the wall protocol, or you have other foods that are anti-inflammatory, so to speak. Anything about that?
Not enough research. I think it's a fascinating area.
Mhm.
I would be surprised if you think about it, think about the meds you're giving your children. You give them a few drops of this and a syringe of that and grind up some pills, but the rest of the time you're eating these large portions of other materials, right? Your diet. And it'd be surprising to me if that didn't have some effect on your overall health, in particular, modulating your seizure threshold to some degree. I would say it's not the most prominent factor in regulating your seizure threshold or these comorbidities, but I think it can help. I always advocate for doing everything you can at the basic level that you have control of, with supplements and diet and sleep and exercise if your child can exercise. All the normal things we think of as healthy, staying away from processed foods, staying away from chemicals, trying to be organic food. Yeah, I mean, it's much more expensive and people are struggling with their budgets, I know, so it's not always possible. But it's something that if there's nothing else, it's not going to do harm. It's possibly going to stabilize the situation to some degree and not cause harm. So I agree with you that we need to do more research on the Terry Wall diet. Super interesting. She's a doctor that was wheelchair bound with multiple sclerosis, and she got herself out of the wheelchair through diet and I think exercise. And she published a protocol. I've looked at it recently because I have long COVID and I have neurological symptoms from long COVID. And a lot of my neurological symptoms are not unlike MS. So I'm looking into that right now. And in fact, I found a doctor online, Dr. Bruce Patterson, that developed a blood panel that is diagnostic of long COVID. And I was shocked to see how much of the biomarkers in that panel were the same that I would do for epilepsy. So he's looking at these inflammatory molecules like IL-6 and interferon gamma and all these other things that are inflammatory that we know have some effect on seizures. So the other area that I'm going to push at this meeting is we're going to have a working group on biomarkers. Can we get blood draws from our kids, our loved ones, get a panel done with, let's say, 20 markers, molecules, and know something about the stage of disease we're in, or if we're being treated, how we're responding to treatment. And we don't know anything about that in SCN8A. It's just a huge gap. And other diseases have this effort on biomarkers. In fact, there's some funding mechanisms in, I think at NINDS on biomarkers that we could actually encourage people to apply for in grants to understand that. But that gets back to your question, Suzanne, because biomarkers, Terry Wall's diet, you'd want to follow your progress because it's not going to be instantaneous, right? It's going to take time. It's a slow go. Any kind of dietary treatment is a slow go, or a supplementary treatment. You got to stay on it for months to see any change. And so we would want to monitor your change with some kind of molecular assay from your blood draw. And those aren't hard to do. So we need to know more about that.
Yeah, and I think that, maybe I could just chime in to share that, that's something that we're looking at, maybe trying to bring somebody in to do a talk with the community about, nutrition, nutraceuticals, those kinds of things, to get some more expert opinion on some of that, but it's something we'll talk about within this community, this meeting as well. And something that's definitely on the radar. And I think your question initially about inflammation too, right? Like that's a huge issue. I think that's emerging. Michael's been saying that for many years, and I think it's emerging more as an issue now that people are looking at. So it's definitely going to be coming up.
So I have a follow-up question, if that's okay. So I've done some training. I've attended a lot of seminars for the Institute of Functional Medicine, and they talk about inflammation, obviously, but then they also talk a lot about mitochondrial function and dietary changes. Again, it's like it's all tied together, but I haven't heard it languaged in this setting in that way. So I just was wondering if you had any thoughts about that, or if you think that's applicable to our space.
Absolutely. Mitochondrial function is critical, and it's tied into all of the things we're talking about. The mitochondria in your cells produce ATP. You use ATP for all the cellular processes in your body. The brain uses most ATP of any organ. Seizures are ATP expensive. So when you have seizures, you're depleted for ATP. It changes not only your ATP, it changes your glucose levels in your blood. It changes all kinds of things when you have a seizure. Knowing more about mitochondrial function and dysfunction is critical to, I think, the research area. And it's something I've been interested in because in our mouse model, I can see that there is a mitochondrial dysfunction that it develops very early. Many of you may have been misdiagnosed or diagnosed as having mitochondrial dysfunction. There are genes that are expressed in the mitochondria that, when mutated, cause epilepsy. So we know there's a direct relationship. Inflammation, mitochondrial function, and all these things operating in the synapse of the neurons are critical and interrelated. So very good point. Again, not something we talk about enough, but something I'm passionate about in our research is what I call the injury cascade. So downstream, if you were to draw a diagram of like what's happening with SCN8A, you start out with the mutation in SCN8A, and then you look at the downstream effects of that, which in the neuronal sense is too much excitatory neurotransmitter called glutamate. And then glutamate, when there's too much of that, it hits receptors in the other neurons that open those receptors up to calcium and to other things that cause the cell to react and to create what they call reactive oxygen species, which in turn cause mitochondrial dysfunction. And it's just a cascade that goes on and on and on. It eventually leads to inflammation and blood-brain barrier dysfunction and all of that. And then you have a vicious circle where seizures beget seizures. We've always known that. There's a guy back in 1881 that said, Gowers, he said, seizures beget seizures. So once you have seizures, they beget other seizures. And we know that there's a molecular component to that, because once this cascade is triggered, it will feed back to cause more seizures. I don't know many people that are working on this, but I do think it's a critical question. If we don't have a gene therapy or a drug that specifically fixes the gene and then cures the disease, we need to look to these other areas for sure. And I don't think we should wait. I think we need to do it now. Only a couple more here. So one of the things we're planning is to do a prospective study, which is part of a natural history study. And we asked, would you be interested in participating? We didn't inform you what it is, but most people said yes. And a quarter to a third said, I need more information before I would know if I would participate. But most of you are open to the idea of participating. We mentioned in the survey a little bit about it, but not much. And I was really pleased to see that people were really open to this, because everything we know about SCN8A right now is retrospective. In other words, as more and more of you have come to our group and we've been able to get data from you, and as the literature has grown and as clinicians have seen more patients with SCN8A, we can say, okay, I've got a 10-year-old patient, I've got a 5-year-old patient, I've got a 6-month-old patient. The registry was completed at these different ages. So we have these time slices, this cross-sectional data of the population. And then we kind of look backward. We say, here's what you are affected like, and here's when your seizure started, here's what medications you took, and we get this retrospective view of the disease. It's very powerful in telling us a great deal about the disease, but we don't really know the way the disease progresses for an individual. All we can do is say, compare the individual to the population, and every individual is a little bit different. It's more powerful to say, let's start today. I'm going to follow you for 4 years, and I'm going to collect data on your situation for 4 years going forward. And I'm going to build some models, and I'm going to make some predictions, and I'm going to see if my predictions come true based on your story and everyone else's story that we're following. And that's going to be a more powerful approach to describing the complete natural history of the study. And it's more relevant to pharmaceutical companies who want to do a clinical trial, because they're going to start on day whatever your child is, and they're going to move forward from that point on. And they want to know if the drug's making a difference going forward. So we've applied for an FDA grant that we're still waiting to hear if we're going to get funded, that would allow us to do a 4-year prospective study with 60 families. There's a limit to how many families we can study, because we're going to study you in detail on a monthly basis. And we're going to give you seizure trackers, and we're going to give you diary, electronic diaries, and we're going to give you headbands to put on at night. And we're going to do all this stuff to really monitor you in a very granular fashion. And then we're going to collect those data, and we're going to see, without giving you a new medicine, that's up to you. We're going to follow your progress and see what happens. And then we have models that we built from the retrospective data, the registry data. We'll test those models with our prospective data. And then in the end, we hope to have described the natural history with the current state of the art of treating the disease. And then when companies come up with new medications and gene therapies 4 years out, they may use our population and our prospective study as the baseline and not have to deal with a randomized controlled wing, placebo-controlled wing of the trial, because they have it already, essentially. So it's a really good opportunity. It's probably the next most important thing to do when it comes to collecting data directly from families. Although every family that comes to us, we want them to fill out the registry, and we want to get data on a retrospective basis. But I think the biggest challenge for our prospective study will be to get buy-in from families for 4 years. Because if people drop out at year 2, that's not going to be very helpful. But why would families want to stay? Why would they drop out? We're going to try and give you enough perks to make it interesting, and give you enough feedback about what we're learning to make it compelling. And then, of course, our meeting structure will be different for the families that are in the prospective study, because we can actually get together on these Zoom calls and talk. And the clinical coordinator would be responsible for reaching out to you, and there'll be a survey that you fill out, a brief survey every month. Did anything change? Meanwhile, we're collecting your seizure data, and we're asking, how did you sleep? Did you take your meds? Did you have seizures? All this stuff. And that's going to be coming to us on a daily basis. So it's very exciting. I think it's going to be very powerful. And that's what we're asking about here. And I was pleased to see a lot of interest. Does it sound like something you'd be willing to commit to for 4 years? That's the question. Because I think we can do it. I just hope that we have, again, the buy-in from 60 families. There may be people knocking on the door going, why can't I get in? That would be a good problem, but hopefully we can get something solid going.
Yeah, I think this community, these numbers are amazing, and it's a commitment, and it's work on your part. But like Michael said, these are the kind of things that are going to shift the number and kinds of pharmaceutical companies that are interested in working in SCN8A. This is a very attractive thing. And so we'll do a lot more education about this if and when, well, also when the FDA grant comes in. I think we'll probably do an educational session for the community to really walk you through the ins and outs of it, so that you can see what it entails. And hopefully people will raise their hands. Yeah. All right.
All right. So the last question was about a clinical trial, and not would you participate, but would you participate for the following reasons? Would you like to see a new anti-seizure medication? Would you like to see a new gene therapy, or you're just not interested in clinical trials? 9% were not interested in clinical trials for whatever reason. Maybe they just felt like there was no hope because they had loss of function and didn't think a clinical trial would be working on something like that. Or there might be other reasons why you might not want to participate in a clinical trial, and we'll talk about those in the next slide. But again, 60% of you would be happy to participate in a trial for a new gene therapy, or at least think that's an important priority. Or about a third of you said, I'd rather participate in a trial for a new medication. Again, those are short-term and long-term prospects. Clinical trials, though, for new ASMs are ongoing for a couple of them. So that's already happening. New gene therapies are a ways off. No, so why would you or would you not participate in a clinical trial? And I separated the responses into what I considered the benefits of participating versus the risks of participating. And so obviously the biggest benefit was I want to improve quality of life, and that's one of the reasons why I'd participate. And things like improving non-seizure outcomes was another reason. So that 9% that didn't want to participate, it can't only be loss of function. It has to be because a lot of you said, I want to see my non-seizure outcomes. Of course, I'm kind of conflating things here because even gain of function have a lot of non-seizure outcomes. So maybe not. Home visit options, those are the things that were benefits. The risks were potential side effects of the treatment and the stability of the current management program. Like, would I have to go off of that? That sounds bad. That sounds risky. And safety risks. So those are the top risks. And I don't think there's a lot of surprise here. This information might be helpful for companies that want to do clinical trials. So I'm glad we collected this information from you guys. Okay, I don't really have any other results from the caregiver survey, but we could talk about anything else you'd like to talk about at this point. And I'll stop the share there.
Well, if I can just jump in about not wanting to participate in the new therapies. Just for us, it's pretty clearly our kid is doing very well at the moment. He's off of medication. This group doesn't know that yet. But he's almost off of medication for 2 years now. He's doing very well. He's developing like a normal child. So why would I risk anything to disturb that?
Absolutely. Yeah, if your child's doing well like that, and I think there are a few cases where I've heard recently, and you might have been one that we talked about earlier, where you're able to take the medication away and be okay. How old's your child again?
It's Fedon. He's 6 now.
He's 6. Yeah.
Yeah.
Yeah. So, I mean, we hope that there are cases where it gets fully resolved on its own. It's not that common, unfortunately. There is a category called SELFie, which is self-limited familial infantile epilepsy. And there are cases in the literature where the clinicians publishing these articles claim that SCN8A does resolve fully in some individuals and that they don't have any cognitive issues. But I will say there aren't many cases like that as far as I know. So you may be one, maybe a few others.
You mentioned it in the last Zoom meeting.
Yeah.
And I think as the landscape for SCN8A continues to expand and genetic testing becomes more common and kids get tested earlier in life at first seizure, or I think we will probably hopefully see more of these kids where it is resolving. You know, we know that they're out there, but I think a lot of times genetic testing is delayed and kids may not get the diagnosis early. And then if everything resolves, orders genetic testing, right? So that we never know that it was SCN8A that did resolve. So I think, you know, in the next 10 years, it's going to change a lot. You know, what we know about SCN8A, given how genetic testing is becoming more advanced. I will say that we're also in the final stages of working with getting a partnership together with a genetic testing company to do free whole genome sequencing with our community. For those who maybe have only had limited testing and need to learn more about whether there's other things going on, and as a research effort, right? I mean, when you can really look at your whole genetic, code as a, the instructions for your body, and Michael would say this in a much more eloquent way than I as a geneticist, but when you can see that full picture, we can start to understand the variety of reasons, or the reasons why somebody who has the same mutation as somebody else is possibly doing better than the other, right? Maybe it's because there's some protective factor in something else in their genetic code, or some other factor that's making things more severe or profound. So, I think there's going to be, on the heels of this research roadmap meeting, which Michael mentioned is coming up next month, there are going to be more and more opportunities for this community to engage in research in ways like that. And we will always be really transparent and put out there, what is going on and why we're doing it, and, what the risks are and what the benefits are. But I think it's a really, that's what's coming up on the horizon, and the engagement of the community in research is what's going to get us closer to better treatments, because the more we know, the more we can share, the more we can have access to grants like at National Institutes of Health or FDA, or private foundation grants. And so I think we're really moving in a really exciting direction, and there's a lot of excitement about SCN8A. We are doing a lot of work to build relationships with pharmaceutical companies and try to get them interested in SCN8A. In fact, we have, 3 or 4 pharmaceutical companies coming to this meeting that don't currently have SCN8A programs, but we're really trying to convince them about how incredible this community is. They show up to do research, they're engaged, they want to contribute. And that's going to be the key to us continuing to learn, is that we continue to engage in the registry, coming to these meetings and sharing and learning, and being really active in the community. So
Tyler's got
Tyler, yeah.
Hey, Tyler.
Yeah,
Hi.
I'm just trying to expand upon one point that Gabby made, but I think I think it could be an interesting direction for research, owing to I referenced it in one of my comments, the Barecki paper back from 22 for SCN2A, which kind of classified these 4 groupings of variants based on their phenotypic variability, which were basically, there were these groupings of ones that were kind of always severe, not like any child that had it was always severe, pretty homogeneous. There was one where it was like effectively always, mild or benign, almost benign pretty much. And then there were these ones that were slightly variable. And then there were these ones that were extremely variable. It was either benign or severe. It could go the whole gambit. And those were a relatively small proportion of the variants, but they seem to cause the channel to fail in unique ways. So that could be something, and 8A has similar analogous variants to 2A. So that could be something to look into in terms of looking at which genetic markers in the background, or which genetic background could indicate or could be quote unquote protective against the full ravages of SCN8A.
Yeah, that's super interesting, Tyler. When you were talking, I was thinking, we know that there's a connection between where the variant is in the channel, which, when you tell us your P dot, we can map that onto the channel. Mm-hmm. And some of those variants are in places that are really unfortunate and change the channel dynamics a lot.
Yep.
And some of them may be on an edge effect where it really depends on how the channel is modulated by other molecules, because we know other molecules bind to SCN8A, to NAV 1.6, FGF12 and PRRT2.
Yep, PRRT2.
Yeah, they're modulators of the channel, and some of those variants are in the C terminus. So like at numbers bigger than, let's say, 1750 or 1790. And then there's other variants that are sitting on splice site. So it's complicated genetically, but when the DNA makes the protein, there are pieces of the DNA that aren't expressed, they're cut out. Like if you're making a film and you're editing out little chunks, and the way those pieces are taken out is modulated by particular sequences at these boundaries. And some of the variants we see are at these boundaries, and those splice site variants are highly variable. some of them cause infantile spasms and severe outcomes, and others not so much. So there's so much to learn. And I think one of the nice things about knowing this is that it gives you some a prognostic view. So if you could on day one say, here's my genetic report, here's my variant, and we could say to you, well, here's what we know about that variant. It might be good news, it might be bad news. That's the you got to brace yourself, but it is information that's highly informative.
Yep.
But anyway, super interesting.
Yeah, and just one last thing before I give the floor back. What you had mentioned about the hypertension, I would imagine you were probably also interested when the Wong lab presented I'd say the follow-up to the Aiken paper on the carvedilol, which is also a hypertension drug, admittedly a different mechanism that they looked at on a 8A mouse model. That was that also seemed to show some sort of protective effect. So
I saw the paper, but I haven't read it yet, so I'll have to check it out. Yeah.
Yep.
The type of drug I'm talking about is been published now in 2 studies, epidemiological studies. So they took many thousands of people with hypertension, and some of them happen to have epilepsy.
Yep.
And they found that some of them taking a certain class of drug called an angiotensin receptor blocker did better with their epilepsy. And that was a statistically supported finding because they had 20,000 people in the study. But it just so happens by coincidence, that's the drug class I'm testing.
Oh yeah. And I recall that from some previous talks. Now it'll just be, once you get the paper published, to convince some doctors to try it off label.
It was just accepted. Yes, 2 days ago they sent me the acceptance letter.
Hooray! All right, I will cede the floor here. Sorry for using 2 separate questions.
No, you're
good to hear from you, Tyler.
Always good to hear from you. Yeah.
All right, I see we've got Matt. He's got a hand up, and then I'd love to do some go around and hear from some families. I know we've got a new family that we'd love to check in with and hear from. So.
I've also got a question for everyone just to think about in the background, but my major concern with the survey, and I think all of us who crafted the survey wanted to make sure that all the stakeholders, all the families, all the loss of function, gain of function families were represented in the survey. And I know we have a lot of families with a loss of function on this call. If you could just chime in every once in a while when someone's not talking and tell me if you think your needs were covered, because we want to make sure that they are.
And if you haven't yet, you can do the survey, which I'll put the link in the chat again too. There's still time. We're going to keep it open through the end of the month to hear from as many families as possible. So
yeah, I just want to know if we're not going to change the survey. So hopefully we've covered it, but we may not have. So I'm really interested in your feelings about that. All right, Matt, was it Matt had his hand up? Yeah, go for it, Matt.
Hi, good afternoon. I just wanted to give an update on Malcolm to you guys. Malcolm's been doing the trial for with Neurocrine Bioscience for 2 years now. If anybody is interested in doing a trial, definitely recommend it. If you guys we were kind of at a point with Malcolm where we didn't know where else to turn. So if you're open to that, I definitely would encourage. We've had a great experience with Dr. Sullivan up in UCSF, and while Malcolm's not completely seizure free, we have seen improvements and benefits from the trial. So it's been a great experience for us. So I would just really encourage people to, if they're able to do it, there is a commitment to go out and travel and stuff, but if you're interested in doing it, I would say give it a shot. The other, I did have one question about when we talk about gene therapy, and I think the last time we talked about it was kind of that's kind of a far off possibility for kids with SCN8A. Is there more interest from companies willing to do something like that, or did something change in that regards? Or because my understanding was that it was maybe more for blood issues, or not necessarily for these for the SCN8A, but I just wanted to see if there was any. And I was kind of late to the meeting, so I apologize for that. But I wanted to see whether there are any like new breakthroughs in that, or is there interest coming for people to explore that, or what? So,
Bobby, you have the best handle on all the different gene therapy companies.
Yeah, so this is really an emerging space. There are not really there are clinical trials happening now in some disorders, especially disorders that are more kind of advanced in the space, like Dravet, which is often associated with the SCN1A gene. So there are some trials in that are happening now, but we just really have not seen it in the epilepsy space yet. So it really is emerging. But there's a lot of interest, as Michael mentioned. We go to a lot of meetings with NIH. I've heard I was at a meeting with the NIH or the director of the National Institute for Neurological Disorder and Stroke, NINS, which is the group within NIH that manages epilepsy. They do a lot of grant making to epilepsy research. And, he is very excited about it. He really does think that's the future. I think he's not alone there. And that's kind of where we're headed. But I think it's probably still 5 years off till we see something, hopefully in SCN8A. And as you know, there are different levels of genetic, things that fall under kind of the genetic therapies, as some people kind of classify them. There's these ASOs, antisense oligonucleotides, and there are ASOs for SCN8A. There are several in the works. These are medications that have to be given every 3 months typically, but they do go in and try to correct some of the misspellings in there. And Michael, correct me if my science is wrong. But those are ongoing treatments. And then there are full like genetic therapies where they actually go in and they change the genetic code so that it no longer has this error in it. And those are one and done kind of therapies where you do it and then like hopefully things go well. And if they do, then whatever the impact is of that treatment lasts forever. And if it doesn't go well, then there's no going back. And so we're being really careful about how we kind of delve into that. But we have a great session on our website where one of our SCN8A parents, Madeline Uden, did an overview of different kinds of genetic therapies and the risk and how they're administered. And I will put that in the chat. It's on our SCN8A Alliance website under caregiver, like recorded caregiver sessions. But that's kind of, where we are now. We're kind of on the precipice of it. We have several companies. We have a company who's coming to the meeting who actually has a successful genetic therapy that they've created in another disorder. And we're really trying to get them excited about SCN8A. And so I think, Matt, it's a really good question. I wish we had a better sense of the exact timing, but I'm thinking that probably in around 5 years, we'll have something that's available.
Yeah.
And Michael, do you want to add to that? Yeah.
I'll just say that our researcher or lab scientist survey, which parallels the one you guys did, asks about these different approaches. And there's a huge excitement and interest in ASOs in particular, as Gabi was saying. These are little molecules that can change the way genes are expressed, higher or lower. So if there's too much SCN8A, you can knock it down. If there's too little, you could knock it up. So it can both handle gain and loss of function. And then the other one you were talking about was CRISPR-Cas9, which is a way of altering the DNA sequence. I think those are further away from being approved because we don't know about all the off-site effects that could have on your genome. But the ASOs seem to be the most promising right now. But there's a huge amount of interest in this. And so we will be summarizing, again, the researchers, the caregivers, and the clinicians, how the different surveys interact. And that we haven't done that yet, but I can report on that soon in our next science update.
And we'll also do an update for the community after the roadmap meeting to kind of share what happened there. We're also going to publish a paper as a result of that. So there'll be an opportunity to really follow along and hear what happened there. And then I promise you there's going to be so many more opportunities for families to continue to share their priorities and their concerns and their ideas. Because, everything that we do, we want it to be really centered on what families want and need, and are seeing in their kids. So
Gabi, I think Matt was also asking a little bit about the industry involvement in gene therapies and how much interest there was in that. Not just the techniques, but the industry people interested.
Yeah, I mean, I think for a rare genetic epilepsy, we have very good interest right now, and that's a huge part to the kind of engagement that this community has been giving to research and the registry and those kinds of things. And there's because there's a very established publication trail on SCN8A that makes it a much more kind of understood amongst these rare epilepsies. But more and more companies are coming into the gene therapy space and looking in the rare epilepsies like SCN8A. And for within these groups, I think that SCN8A is kind of in the top 10-15% of groups that are being looked at. You know, there's a lot of right now, the rate of new epilepsy disorders that are being genetic epilepsy disorders that are being discovered is pretty quick. You know, every month there's a couple new disorders that are being discovered. So there's several hundred genetic pediatric onset epilepsy disorders, and SCN8A is one of the more established. And you know, there are some papers that quote and use SCN8A as an example, saying this is a model community, how you work together with patients and researchers and clinicians to get things done. And, you know, we're really, we're seen as a very attractive community in a lot of ways. And I think that is the kind of thing that's going to bring more companies in and interested. So, you know, I think if there was a number right now, I would say that between 2 and 5 groups of pharmaceutical companies that are showing interest in SCN8A and the genetic therapy space. That doesn't mean they're in it yet, but they're interested in it. And we're really working to push them to say, you should really do it.
That's great to
So,
hear. And I just also want to commend you and Dr. Hammer. You guys do a great job with these surveys, and like you're talking about, pushing to get an established, you know, credible type of science behind it and get these companies interested. So I applaud you guys both on all the work that you're doing. I really appreciate it. And it'd be great. Yeah. If that's something that keeps moving, I'd love to be updated on that as well, or be in the loop. And if anything you have to share on that, we'd love to hear it. So thank you so much.
Thanks, Matt. Appreciate it. And I mean, we have such an incredible community. It's pretty awesome to be able to work. I think Michael and Jayetta and I all feel very lucky to get to kind of work with and on behalf of our community. So thanks for showing up and continuing to show up. It really makes a difference. I want to invite others to come in and share what's going on. Sean, I saw you wrote in the chat about you guys are a new family with Alexis. If you're willing to come on and share how these meetings often work, is that we get an update from families about what's going on. Michael, you know, tends to ask a few questions to help him better understand the onset of the disorder and what it's looked like, kinds of seizure types, what kind of mutation you have. And it's, you know, if you're willing to share the mutation, then we can look and say, is that a common one? Do we know a lot about that one? Is it an emerging one? And then, you know, you can ask questions and we can try our best to try to help and answer. So I'll invite you, Sean, if you're interested, you and Jennifer, to chime in and connect now. Otherwise, you're very welcome to just keep listening. If you're not ready to do that yet, that's totally okay as well. And you might be in the middle of something. I know with our caregiving, we might be changing a diaper or doing laundry or running to do something. So, so Sean, whenever, you know, I see you might be busy now, but feel free to just raise your hand if and when you're ready to chime in, and we'd love to hear from you. And you can also email us. I put both Michael and my emails in the chat. You should feel free to reach out anytime with questions. All right. Anyone else want to check in and say how things are going? Oh, okay. Here, I can, 1 second. I'm pushing a button that might help you, Sean, if you're trying to unmute and it's not working. Okay. Álvaro, ¿qué tal?
Hey, bien. Greetings from Spain. We are at night here.
Yes, and you had a very good
Oh.
soccer match. Congratulations.
Yeah, thank you. And also tennis. and many
And
things. We are
tennis.
in
I
the
was
Olympics.
watching both.
Yeah.
We watched both. They were very good. Nadal and
Thank you.
Alcaraz. Yeah, it was excellent.
Yeah. First of all, thank you, Michael, and all the team, because the survey is amazing. I think it covers a lot of our concerns. It's very complete. So thank you. Thank you very much. And I would like to introduce a kind of a controversial issue, and it's about the all we know that seizures are a central subject in this process, but I'm worried about how do we are sure about we are suffering seizures, objective seizures. I mean, with Ada, with our girl, we are being here in a very big hospital in Spain, in Madrid. It's a very good hospital. And in these 2 months, the last 2 months, they think that maybe some movements of the eyes or of the hands, they are not seizures. They are paroxystical movements. And this is also more complex because of the anti-seizure medications produce this kind of movements. They can make confusion in the EEG test. So now we are going to have new tests in a different city in Spain, in Barcelona. And my concern is about, okay, we talk about seizures, but as parents, and I'm a doctor, but this is not the typical thing. I think families, they are civilians, let's say. And there are patients that suffer seizures very, very clear because they are tonic-clonic. They last for 1 minute, 2 minutes, but I'm sure many families here sometimes have little movements or little symptoms that maybe their neurologist can diagnose as probable seizures. The EEG, it's very complicated to interpret, and I'm worried about if we do research and we base research on something that maybe it's not so precisely determined. Now, I don't know if do you understand what I mean? Because it's very important. If we don't measure properly what we want to improve, it's very difficult. And in fact, if we are talking about gene therapies, new medications, clinical trials, and it's very, very important that we engage the industry, the pharmacy, the clinicians in make the best diagnosis about seizures possible.
Yeah,
sorry
no,
for my English because it's
no, thank you for I know it's a challenge. You're doing much better than I would in Spanish, so thank you so much for your comments.
My effort, yeah, my night effort.
Yeah, thank you for your A for effort, and I'd give you an A- for English, but it's very good in Spanish.
I have something, Michael, I wanted to say on that too.
Okay, go ahead, Sandy. Why don't you just jump right in?
I don't know if this is exactly the point he's trying to make, but I do see, like with Ethan and his Ethan has chorioathetoid cerebral palsy movement, so he's jerking, and that has to be completely separated from when he has a seizure. And I have also been wondering if different movements that the children make are being interpreted as seizures or called a seizure, even though it's actually not a seizure, or it's just the abnormal brain. So with this chorioathetoid movement, we have to control that, or else he's going to, it's like he runs a marathon 4 times a day. So if that's not controlled, it's very dangerous for his heart. Okay, but that's different than when he has, let's say, a fever and then he gets this full-on seizure. The EEG usually only was showing an abnormal brain. It wasn't doing what the normal EEG test is for a specific type of seizure. But I just don't think they can actually measure that at this time.
That's the point. Here in Europe, we have two current, let's say, in Italy and France, they tend to treat the EEG, let's say. They want to make a very clear EEG, okay? But in the northern countries, in Norway, even in Spain or Sweden, they treat the clinics. They don't mind the EEG. So
When
I
you
don't
say
know
clinics, what do you mean? Alvaro, what do you mean by clinics? They treat the clinics.
Clinics the same symptoms.
Oh, okay.
So
yeah.
they don't mind the EEG so much, and they look at the child.
Yeah.
Do they do something with the eyes, or can we gate better, or so? And the point
nowadays, excuse me for interrupting, but we're also treating the gene. So you have the old-fashioned way of treating the seizure semiology, the seizure symptoms. Then you have other doctors that prefer, like you're saying, it's very interesting that you have that split in Europe between different countries are doing it different ways. We see that all the time in the way the clinical practice affects our families, even access to tests. It's very different for every country. But just getting to your question, I think seizure types is one of the most difficult things, not only for families, but for clinicians. And I'm shocked at how well our registry data match clinical notes. So maybe it's because the doctors and the families are speaking well, but we have some data from just clinical notes, and we have data from you guys from the registry. And when we look at the seizure types, they're pretty spot on for both. I mean, they kind of agree with each other. So I think in general, we're catching the big broad brush strokes correctly. But I think when you get down to those small tremor-like movements that could be seizure-like or seizures, and they're not being detected by EEG, because EEG is not perfect, right? It only takes the surface of the brain. It can't see what's going deep in the brain. So EEG is just one different piece of circumstantial evidence. It's all in the court of understanding these seizures. We have this circumstantial evidence, and what the parents recognize as unusual really counts for a lot. And when we do our data in our prospective survey, in our prospective study, we're going to ask families on the seizure tracker to say whether it's seizure type A, B, or C, and then we'll make sure we'll get the symptomology of each of A, B, and C and try and map it to a seizure type with a clinician's approval of that, right? A clinician like Dr. Schreiber will look at each one, a video, and corroborate whether it's a seizure and what A, B, and C are. But turning to the treatments of those different A, B, and C, it's important to know the seizure type, but it may not be the ultimate piece of information, because a lot of our patients have multiple seizure types. I think knowing whether your child is having seizures or not is huge. You know, like for some of the loss of function or mild gain of function, if they're having absence seizures, that's a different medical treatment than a gain of function, a bilateral tonic-clonic seizure. But a lot of the seizure types, focal seizures, tonic seizures, all these other ones, and myoclonic, there might be some medicines that are directed towards each one, but it's not perfect by any means. There's overlap, and you probably know that, Alvaro, as a doctor.
The point is that many of these medications have secondary effects that simulate symptoms like ataxia, or tremor or things like that. So my concern is about how many childs have over medication. Maybe I don't know. It's very difficult, very very difficult. But maybe in the long term, with your registry, we can check those child that are polymedicated with many medications. Maybe they share some symptoms that are not produced by SCNH, but it's a secondary effects of 4 medications shown together.
Yeah.
Yeah.
Absolutely. And we ask you guys about medication combinations, but we ask the clinicians in their survey about polypharmacy. Like, how important is it to find the right medication, and how important is it to know how to balance different medications and when you've gone too far? And I think that's a huge question. And we see all the time. And this is something that also differs by the country of origin. In some countries, the doctors are like just going to add and add and add. And by the end of the day, you've got 5 or 6 medications. And other places, you see they're very lean on what they're prescribing. And they try and add, wean. And in the end, you've got 1 or 2. And having being simpler and fewer is so much easier to see what's making a difference. So we always recommend reduce the number if you can. But again, that's such a difficult area.
This had been a very big step of your consensus, because now it's so clear that you have 1, 2, or almost 3 medications to start with the gain of function or a loss of function, and not to try to use 5, 7, or try everything that you have on your hand.
Yeah, exactly. You've put your finger on a very important area. And I think this is something we can take to the meeting next month and really emphasize how important this is for the researchers, the clinicians, to understand polypharmacy and how you treat, whether you treat the seizure, you treat the symptom, you treat the gene, like where are we on all of this?
And balancing all of that with quality of life, right? Like we want our kids to have seizure control and to have better, but we also want our kids to have quality of life. And if they're on 4 and 5 medications, they're tuned out to the world because they're just, like you said, over medicated. So really, really important point. It's definitely something we'll bring to the meeting. I want to come back to you, Sean, I know you were heading to your settings. There you are.
Yeah, I got to figure it out.
Welcome. Good job.
Thank you. Thanks for having us. We're happy to be here and have found a lot of resourceful information throughout the website and all the research and efforts that you guys have made to kind of modernize it with a better approach for families to understand and interpret and share and communicate with their treating physicians and clinical teams. So thank you both, all of you, for all the work that you've done up to this point.
Thanks, Sean.
Thank you, Sean. Yeah, I think we did meet Sean earlier, and his daughter Alexis.
Yeah. So
I know the variant, and we got some information, but I'd love to catch up with you at the next, I don't know if it's going to be a caregiver meeting, a newly diagnosed, whatever our meeting schedule will be. I think we will modify it in the month of August because of this roadmap. And Gabi, do you want to say a few words about that?
Yeah. And Sean, if you have questions, you're welcome to chime in with them, because I know things are evolving. But you basically, you've seen our meeting structure, and we keep trying to evolve for what works best for families. We've done them by variant. We've done them by where people live. We've done them by language. We've done them by, kinds of symptoms or challenges that people have. So if you have thoughts on, who do you want to be hearing from and what do you want to be talking about in the meetings, just let us know, because I'm going to put out, a question to the community to ask that too. But we want our meetings to be really responsive to what you want to know and hear about and who you want to talk to and be in community with. So we're taking August to regroup, and we'll start again in the fall, with some renewed ideas based on the roadmap and then what you all share. So anyway, Sean, I wanted to make sure you didn't have any questions now. And if you do, feel free to chime in. And how's Alexis doing?
She's doing all right. It's just obviously been a whirlwind, and it continues to kind of evolve. So it's been a struggle to really get a handle on
Yeah.
her progression of things as they're presenting. So nothing particular in the moment of a question. More
Okay.
so just wanted to get some of her genetic coding out there for you guys to take a look at for where she falls within the genome sequencing for that. And, if you guys have an opportunity to take a look at that to see if there's any suggestions of a different medication or a combination of medication that could possibly be beneficial. I know her neurologist refers to your guys's community quite a bit for the pathway of treatment. So it's been overall a pretty good experience, but just trying to find something that's going to give her some better quality is the main goal.
Yeah. Yeah, thanks, Sean. I remember now. Yeah, we saw Alexis on the screen last time. Yeah, she was really cute. Yeah. So, yeah, I mean, if you've sent over all the, you emailed the genetic information, right? We can definitely take a peek at that, Michael. Yeah. Right.
Yeah, I know where his variant is. It's in the inactivation gate, and that's generally treatable with sodium channel blockers fairly well. But details are specific, to everyone. But we can talk to you more about it, Sean, as we hear about how she's doing. She's very young, right? She's 3 or 4 months old, so early
Yep.
on. Early on,
Yeah.
yeah.
Yeah,
Okay.
so she had her first major clonic seizure at 3 weeks old. And then it's kind of just progressed after that to, some daily seizures and then prolonged status epilepticus. And we, you know, she has chorea movements, dystonia movements, myoclonus jerks. So combine them all, and it gets very difficult to decipher. Like the gentleman before was just talking about, a lot of these smaller movements. You can see their eyes go up quickly, and they're making different movements and symptoms that mock a seizure, but the EEGs don't really pull that out of the artifacts that are coming from certain activities on the EEG. But deciphering through that stuff and trying to figure out at times for us when we're at home, like, is this a seizure right now? Is it a different type that she hasn't presented that we're familiar with? So has the seizure ended? Because we've seen on the EEG, it's gone on longer than we thought it may have been going on while at home. And they have a difficult time actually of seeing the full start and full end with the different movement disorders kind of laying over the artifacts. So,
yeah.
Sean, has your doctor been pretty good at answering these questions? Because these are really good questions to ask.
Yeah, we've had really great experience with, she has multiple neurologists through U of M that work as a team, a PMNR, and they're all working together, neurosurgery, for other palliative care, to help, come up with solutions and give us a pathway that's going to work best for her.
That's great. That's good to hear.
Yeah.
But yeah, definitely we'll look to provide, we'll look to join the next caregiver meeting, so we're not taking up time on this call that's dedicated to, the roadmap forward and the research and development. So
right.
Yeah,
Okay.
good
We'll look
to
forward to
hear.
hearing from you. Yeah. Anybody want to comment on the survey? Just maybe Deborah, how about you? Do you feel like we covered some of your needs in the survey pretty well or not?
No, I think it was pretty well-rounded, and yeah, I think it was. I think the question about the clinical trials, I might have even misunderstood that it was just for people with seizures. Because I know with Caden, we've tried different medications for behaviors, but nothing's worked. And it's kind of a scary realm for us because he's had really negative effects from that. So I think that's kind of more like on that. But yeah, I think it covered everything pretty well.
That's huge, a huge gap. I mean, the non-seizure outcomes, the behaviors, the medications for these things, it's a huge gap. So we really are going to try and push that at the meeting.
Yes, and it's tough because we're thinking about behavioral medication, but when you tried it before, they're just like, he just reacts differently. I know one, he went to a psychosis, which was really scary, where he was hurting himself, and it was just, that was a Tenex and Keppra when he had the abnormal EEGs, but never a clinical seizure. So that's been very interesting with like different medications and stuff like that. So yeah, just the side effects of everything.
Yeah,
that's a big part.
definitely.
Mhm.
Jennifer, you also have a loss of function son, right? Do you have issues with, well, going back to the survey, did we get the questions right? Is there something you'd like to push on research for whatever challenges you may be facing? If you're there, maybe.
Yeah, I think for us, like we're still, even though we've had the diagnosis for over a year, we have had zero progress on actually getting any sort of genetic counseling or neurology appointments since the original ones last year. And so the EEGs, they saw like pre-seizure activity and like artifact and stuff like that. But like, I think what the other people are mentioning about the different movements not being seizures, but like maybe CP related or some, because that stuff has been mentioned to us as well. I feel like where we are at, like geographically, our doctors around here are not really understanding, or like they're like, oh, well, as long as he's doing okay on a daily basis. And it's like, but he's not. Like when you look at like his PT report from last year versus this year, the gross motor delay, like significant backsliding. So it's just hard to advocate for him when people are just like, well, he seems healthy, he's doing okay, and it's like, but he's not. Like his apraxia is getting worse. Like he's still not able to communicate very well or clearly. But they chalk it up to a behavior situation. Oh, that's his autism. That's his scripting from his, from autism. And it's like, but then I have his psychologist is like, well, I don't think he's autistic at all. I think what's going on is completely related to his cortical dysplasia and the other stuff going on, because he has the compassion and he has the ability to connect with peers emotionally and stuff like that, which I don't know. So nobody can seem to get on the same page with us.
When you say apraxia, do you mean speech or other movements or other executive function type stuff?
Speech, as it's been explained to me.
Okay. Yeah. All right. Yeah. Speech is one of the, as you saw in the challenges, it's one of the #1 challenges. And for the milder cases too, speech is the last thing to be sort of come on board. So yeah, speech is a complex phenotype, if you will. It involves a lot of executive function, a lot of fine motor control. It's tough, and we take it for granted, obviously, because it's we do it early on, but it's got a lot of pathways that have to develop normally to get it done.
Right.
And I think the only thing you can do about it is to do speech therapy. I mean, you can't take a pill for speech, but maybe there's something related that could help, but we don't know what that is.
Yeah, unfortunately, in the area that we're in, he because he's school age now, he does not qualify for clinical speech, and their school district being where they are, they rely on virtual speech therapy, which for a child who has ADHD diagnosis, autism diagnosis, apraxia, whatnot, and then to only get speech support virtually once a week, it's not ideal at all.
Yeah, definitely.
Michael,
Yeah.
I know we're running out of time.
All right, go for it.
I just want to say one thing. So Emily, I have a daughter, she has a loss of function, so she's high functioning, with a lot of behavioral issues, low IQ. She's in a group home in Iowa City. And now Ethan has the gain of function and loss of function, and he's severe and profound. So what I do find interesting is that for some of the non-seizure behavior for both of them, it took a while for Ethan to get the chorioathetoid cerebral palsy diagnosis. They really wanted to make sure it's not a common one. But then with Emily's behavior, they're being treated with the same drug for some of those non-seizure issues. And it's working for both of them.
What is the drug?
It's the risperidone. It's psycho whatever med for people with brain issues, with.
What do you see in Emily? What do you see improved?
Oh, her entire ability to associate with anybody, as opposed to living in a fantasy land like a 12-year-old girl and make things up. And just, she has this very young, yeah. But then with Ethan, it helps with his movements that are uncontrollable.
Well, that's
so
super.
yeah. And now Emily, my daughter, is 33. She's going to be 34. So I've had all of that experience with her. And then with Ethan, this is Emily's son. Because of some of her issues, she had a son. It was fine, but he has both loss of function and gain of function mutations. And it did take a couple of years to really, and I'm talking to Sean right now with your 4-month-old, it really just takes some time and observation and for them to come up with like the right thing.
Well, I think that drug is it's on the serotonin pathway, reuptake inhibitor. So it's an antidepressant, but it's also it's a neuroepinephrine reuptake inhibitor as well. But in any case, fenfluramine is also on the serotonin pathway, and that's one of the off-label drugs that people are interested in for seizures. So it is a drug that may be one of these things that can have both behaviors and seizures. I don't know about the chorioathetoid movements, but that whatever you're reporting is super fascinating, and it'd be great to learn more about it.
Any other thoughts or any other topics that we didn't talk about that you really want to make sure that doctors and researchers know is an issue in SCN8A? I mean, thank you for sharing, Deborah, those issues, because I think we're having this it's emerging as more and more of a challenge around the behaviors and the, access to the right treatments and correct, diagnoses even around that. So that's a big one that's coming up. And we actually have a epidemiologist specialist who's been studying work in these rare epilepsies and these rare disorders for, I don't know, 25 years now. And she's going to be at the meeting with us, helping us think about how we frame these needs based on that survey and think about how do we start working to do more research in those areas. And I think in some cases, it's going to be very, very SCN8A driven. And in some others, it's going to be, how do we do this with partner organizations who are, who have really similar, like communities. So for example, our partners from SCN8A will be at the meeting there with us to contribute and listen. There's a lot of similarities between our disorders. They are the ones that we actually did that webinar on behaviors with very similar populations. And, when it comes to things like sleep and GI issues and behaviors, those are things that are probably not directly associated with SCN8A, but with disorders like SCN8A, because of, the brain chemistry and a number of reasons, a lot of our kids have those issues. So there's going to be opportunities there to partner with different, groups to make sure that we, frankly, it's expensive. So, we as a community don't have endless funding. So, it's an opportunity for us to learn together, and use these specialists in a really smart way so that we're learning across these disorders together and finding answers. And that's going to be the challenge too, is that I imagine leaving this meeting, we're going to have a lot of priorities that we know are coming. And just so you know, everything is going to be focused on family priorities. Clinicians are going to have their priorities, researchers are going to have their priorities, and those are great, but we're always going to bringing it back to, well, families want this. How do we make sure that we're addressing what families want and need? So the challenge is going to be, we're going to have lots of great ideas, but we're going to have to find money to pay for all of these, these great research opportunities. So, we're going to be doing fundraising within our community, and we do not like asking families for money, but we are always, happy. There's plenty of people who give on a monthly basis or give when they can, and we're grateful for that. But, we'll be hoping that if you as, families can help us think about other ways we can do it, get funding for the community, if it's a bake sale, if it's a lemonade stand, if it's, I know somebody who's, got a lot of money and really likes my kid and would always want to ask how they can help. Maybe that's somebody who'd be interested in giving a grant to help us, learn something. So it's going to take all of us working together to, thinking about how do we really find the resources here. We keep waiting for a really rich SCN8A family to show up so that we, and they'll want to help pay for stuff, but, they'll come in time. And, in the meantime, we are continuing to really push this forward and are really excited. So I think, many of you have already been contributing from in the registry, which is really our long-term way of understanding the disorder and tracking and learning. This prospective study that Michael mentioned is going to be really exciting and help us move the field forward. This research roadmap meeting is going to be a critical next step for us. People are really excited about this meeting. It's really exciting. So, it's happening on a weekend, and we've got doctors and researchers and pharmaceutical companies who are taking their weekend away from their families to come and be a part of that. So I think there's really going to be some exciting stuff coming out of that. And we'll, of course, keep you updated and share more and give you opportunities to weigh in and give us feedback, and continue the conversation. So I'm trying to keep up with the chat. I know there's a lot going on here. If there's anything else that people have put in the chat that we haven't addressed yet, feel free to chime in the last few minutes here, or if anybody has any thoughts. Yeah, Tammy.
I had a couple parents say they would like to have it brought up about the medication levels being checked for their children, because a lot of people, they struggle to get those done. And then the taking seizure medication away and causing seizures in a hospital setting is still dangerous for children with SCN8A. That was a concern for two parents that I spoke to. So, fighting to get that medication level, they're saying that they're at an adult dose, but their child is metabolizing the medicine so quickly that their level in their blood is not, where it should be still. So,
yeah. No, those are really important ones. Thanks for mentioning those, Tammy. Tammy's always got the pulse of what's going on in the community. Tammy and Megan and our
kind of, when my life isn't so crazy all the time.
I know how that goes. Yeah. Thanks. One of the other things that we're doing, and if anybody's interested, is that we are, we also are working not just in the room that day or for this meeting, but looking to spread awareness about SCN8A. We are always looking to share family stories and experiences, and so I'll put in the chat. This is a form where we just ask a little bit about your kid, and we'll be making these really beautiful posters for the meeting to hang to tell a little bit about the different experiences our children have. So if you're interested in sharing your kid's journey and story to help inspire these researchers, or maybe for us to share on our social media to really raise awareness about the disorder, that's a form with these new posters that we're looking to start building out. I can share, like, it's going to look a little bit like this, but not exactly. Oops. I'll just bring it up real quick so people can see in case you're interested. This one's not done yet. I was trying to make one for Elliot, but I didn't get very far yet. So kind of like this. And we want to know a little bit about the child, what, quality of life looks like, what their favorite things are, how do they communicate, what does it mean to be a caregiver of someone with SCN8A? So, it's really going to be, I think, a great way to share more about our children and what our challenges are and what makes them so special and wonderful. So, all right, I'm going to stop talking, and Magdalena, please go ahead.
Hi, everyone. So I had a question. Our little girl is she's just turned 1 year old today, and she's actually worse than she was like 2 months ago, because at a certain point she was crying a lot. Then after a few days, we figure out that she has a virus that was probably causing her pain, and after that, soon after that, her tooth came out. And then today, in the meeting, when Dr. Michael, when you mentioned that kids can have more seizures when they're eating, I'm actually trying to connect this with the question that Gabi asked about what would we like our clinicians to be more aware of, or something that we could communicate, because I feel like our doctor, or I mean her doctor, is kind of reluctant to make any connections to her seizures other than medication she's on. So whenever I would say well, she had this virus, maybe that, or the pain that it caused, maybe it made her more susceptible to the seizures, she would just roll her eyes almost and be like, no, we have to change something in the medications and stuff like that. So especially the teething part, I feel like if I would mention that she's teething, she would be like, are you nuts? So what is actually the source of that? Could I in some way communicate to her
you
some
can
of
use the
those
word
things?
inflammation. Yeah, I think doctors should know that inflammation and seizures interact. So teething it can cause inflammation, virus, vaccinations, these all things can lead to some inflammation that will reduce your seizure threshold. So doctors should know that. It shouldn't be considered conspiracy theory or whatever.
Yeah, I just feel like it's she's less prone to if she has worsen, if her seizures worsen, she is much less likely to hold it out, to wait with the changes than she should, I guess. I don't know, because she's just 1 year old, and there are some things that we haven't tried. So I feel like a part of her worsening now is because we made so many changes when she was actually pretty low with the threshold. So I don't know if we did a wise thing.
Yeah, no, I mean, we could talk more about the particulars at some point, maybe at our next call. But yeah, I mean, hopefully your doctor knows that when you try a new medication, you have a period of time where you have to let it work, come up to full dose. Our kids do need higher doses than typical. But letting things come through and give it time to work before you decide it didn't work. And if there's too many changes going on, we don't know what's working, what's not working. We don't know if that makes seizures worse. I think it would. I think anytime you withdraw a medication, it's putting your system at risk for more seizures. So it's a dynamic process. That's what the clinician should be trained in. So hopefully they are aware of that.
Okay, thank you.
Yeah. But I mean, I could try and find something that a publication showing the relationship between inflammation and seizures, if she really is. I just think that's not going to be an ongoing battle you're having with her, because inflammation, it comes and goes. And as Octavia gets older, you're going to have the teething won't be an issue anymore, but there'll be other things like vaccinations and other things you should be wary of during times of seizures. You don't want to cause more inflammation during a time of seizures than you have to. So you might have to relax the vaccination schedule. That's something to be aware of. And hopefully your doctor will roll her eyes at that, but some do.
Yes, thanks.
Yeah.
Thanks for the answer. And if you dig something up, please do share.
Okay. All right. I will. I'll look for something.
Yeah, good luck on Googling that. We'll share anything we find, and hopefully things get better. And happy birthday, sweetie. All right, we're just about at time here, but if anybody else has any questions or anything pressing that, we're happy to chat or connect, but hopefully you always know how to find us. I will put again the emails in the chat for Michael and I. And we love hearing from you, so please reach out anytime. And
Something already in the on teething at the Epilepsy Foundation, teething and seizures. So hopefully, I haven't read the article, obviously, but it might have some information that be useful for your doctor.
yeah.
So he's not alone in that. My son's 7, and we still run into that issue when he's ill, or if he gets hurt, and we go into the ER and he's having body movements that I feel are seizures and whatnot. I do get pushback from doctors of why that would be a part of an injury or an illness, and it's like, well, he has SCN8A. So I do still struggle with advocating on that monitoring for Dolan too. And it's like, if I had something to hand to the doctor that says, yes, this child has this genetic diagnosis, and we need to monitor for this in times of injury or illness, so that we can capture seizure versus other activities going on in the brain and body. I don't know if that makes sense, but I have felt that way as well, where the doctor's like, oh, well, just give him Tylenol, he'll be fine. It's like,
Yeah.
Yeah, I mean, I'm curious if you would say a little bit more about what would be helpful to hand over, like what kind of information. I mean, we do have, like a 2-pager from the consensus that's on the website that is kind of a summary of all the different phenotypes and what we learned from that about, how kids need to be treated, all these kinds of things. I mean, what kind of information would be most helpful to have available?
yeah, the longer information was hard to share with the doctor. She was like, oh, when I have time, I'll look at this, but it was never perceived. You guys do a really great job with like infographics. So maybe something that would say a child who is experiencing a medical emergency may be impacted differently because they have SCN8A, and kind of like a flow chart of, okay, this child has this genetic diagnosis. This is happening with the body because of injury or because of illness. I don't know how to explain it, but I don't know if there's I'm sorry, but we live in a really rural area. Baby doctors are here. It's July. It would just be helpful to have something very concise that says, yes, this is a red flag that you need to actually consider, and not just a parent being overprotective or hypervigilant or whatever. So
Gotcha. Thank you for sharing that. Yeah.
Yeah, we can create, so we'll try to work on some stuff and we can, yeah, we can send it out to people to get feedback on too. Thank you. All right. Anything else? I think this has been a really fantastic discussion. Thank you, everybody, for joining us, for listening, for contributing, for being just such active and amazing members of the community. That's really what's driving us forward and making it possible for us to have meetings like the roadmap that's coming up, where we're able to engage, a wide variety of doctors and researchers and pharmaceutical companies. And we'll look forward to giving you guys an update in the fall. All right. Reach out anytime. I hope you're doing well. Enjoy the rest of the summer. Good to see so many of you. Take care.